right, so we need to talk about how the brain is set up. What is the actual structure? What's happening there? What are the tissues that we find? And how is it basically constructed? So that's what we are going to talk about in this lecture. So there are two types of matter. We call them gray and white matter. Gray matter is basically the seat of neuron cell bodies, dendrites, and synapses. It is a dull white color when fresh, and that is because it doesn't contain a lot of myelin. It forms the surface layer in the cortex over the cerebrum and the cerebellum. It also forms the nuclei deep within the brain. Then we have white matter. These are composed of bundles of axons. It lies deep to the cortical gray matter, and it has an opposite relationship in the spinal cord. It is a pearly white color due to containing myelin. And we know that myelin basically is what envelops the nerve fibers. It is composed of tracts, bundles of axons, that connect one part of the brain to another and to the spinal cord. Okay, so gray and white matter. Then we have what are called meninges. Meninges are three connective tissue membranes that envelop the brain. They lie between the nervous tissue and the bone. We see these same meninges in the spinal cord, and we call them the same names, which are the dura mater, the arachoid mater, and the pia mater. They protect the brain and provide a structural framework for its arteries and veins. So the first part, the dura mater, this we find in the cranial cavities, and it comes in two layers, the outer and the inner layer. The periosteal is equivalent to the periosteum that we see in the cranial bones. And then you have an inner lining, and this continues into the vertebrae canal and forms a dural sac around the spinal cord. You also have a cranial dura mater, which is pressed closely against the cranial bones. There is no epidural space here, and it is not attached to the bone except around the foramen magnum, the sella tertia, the crista galli, and the sutures of the skull. These layers are separated by dural sinuses, which collect blood that is circulating through the brain. The dura mater basically folds inward to extend between parts of the brain. And it lists here where that is happening. Okay? The arachoid mater and the pia mater are similar to those that we saw or will see in the spinal cord. The arachoid mater is a transparent membrane that we see over the brain's surface. And the subarachnoid space separates it from the pia mater, which is below. The subdural space separates it from the dura mater, which is above in some places. The pia mater is a very thin membrane that follows the contours of the brain and even dips in at the sulci. It is not usually visible without a microscope. Your brain also contains ventricles, and there is also something called the CSF, which is the cerebral spinal fluid. The ventricles are 4 internal chambers that we find within the brain. There are 2 lateral ventricles, one in each cerebral hemisphere, and there is a 3rd ventricle, which is a single narrow medial space beneath the corpus callosum. There is also what's called the 4th ventricle, And this is a small triangular chamber that we find between the pons and the cerebellum. We also have a choroid plexus, which is a spongy mass of blood capillaries on the floor of each ventricle, and an ependyma. This is the neuroglia that lines the ventricles and covers the choroid plexus. And this is where the cerebrospinal fluid is produced. So what is cerebrospinal fluid? Well, it is a clear, colorless liquid that fills the ventricles and the canals of the central nervous system. And the reason that we have it is because it aids in protection and filtration. Your brain produces and absorbs about 500 milliliters per day. The production begins with the filtration of blood plasma through the capillaries of the brain. The ependymal cells then modify this filtrate so the CSF has more sodium and chloride than plasma, but it has less potassium, calcium, glucose, and protein. Your CSF continually flows through and around the central nervous system. This is driven by pressure and the beating of the ependymal cilia. And there are also pulsations in the brain that are actually from your heartbeat. The CSF is secreted in the lateral ventricles and flows through the intervertebral foramina into the third ventricle. It then flows down the cerebral aqueduct into the fourth ventricle. The third and the fourth ventricles add more CSF as it goes through along the way. So it's a continual addition. And a small amount of the CSF fills the central canal of the spinal cord. And then the CSF is reabsorbed by the arachoid villi. What are the functions? Well, I already mentioned protection. It also deals in buoyancy and, as I said, filtration or chemical stability. So it allows the brain to attain a considerable size without being impaired by its own weight, kind of like it's floating. If your brain was just rested or just sat basically on the floor, it would be so heavy that the pressure would actually kill the nervous tissue. So this cerebrospinal fluid allows it to be buoyant and not so heavy on itself. It also deals in protection. It protects the brain from striking the cranium, the bone, if your head is jolted. You may have or may not have hit your head hard once in your life, and your brain didn't explode when it hit your cranium, your skull, and that is because there is this fluid that's kind of acting as a middle ground or a barrier. It does mention here that there is something called shaken child syndrome, and this can occur if a baby is shaken very hard. It can cause a concussion. The CSF also functions in chemical stability. The flow of CSF rinses away metabolic waste from the nervous tissue and homeostatically regulates its chemical environment. Your brain has a supply of blood. Your brain is only about 2% of the adult body weight, but it receives 15% of the blood. And this is because neurons have a high demand for ATP and therefore oxygen and glucose. So we need a constant supply of blood to the brain to help to make sure that our nervous system is able to work as efficiently and as fast as we need it to. So if you have a 10-second interruption of blood flow, you could become unconscious. A 1 to 2 minute interruption can cause significant impairment of neural function, and 4 minutes will most likely cause irreversible brain damage. So very important to have that constant supply of blood to the brain and to all the neurons and our nervous system. The brain barrier system. The blood is also a source of antibodies, macrophages, bacterial toxins, and other harmful agents. The brain barrier system strictly regulates what substances can get from the bloodstream into the tissue fluid of the brain. So basically, the brain barrier system is controlling what is going to be able to get into the tissue of the brain and the fluid of the brain, because we have to be highly selective as to what is going into our brain and into those neurons, because they are basically controlling our entire body. So we need to make sure that not just any old chemical is getting in, right? So the blood-brain barrier protects blood capillaries throughout the brain tissue. Okay? And through these tight junctions and these different neuroglial helper cells, everything is regulated and sealed off so that only certain things can get in. So you have this brain barrier system, right? That's exactly what we're talking about. And it is highly permeable to water, glucose, and lipid-soluble substances such as oxygen, carbon dioxide, alcohol, caffeine, nicotine, and anesthetics. And that is why you feel an effect if you have these items, right? If you ever drank alcohol, you may feel a little tipsy, and that is because it is able to cross this blood barrier system. Same with caffeine or nicotine. You get kind of a jolt from those things, and that is because those chemicals are able to somewhat interact. The BBS is slightly permeable to sodium, potassium, chloride, and the waste products, which are urea and creatinine. This can cause an obstacle for delivering medications such as antibiotics and cancer drugs. Trauma and inflammation can cause the BBS, can damage the BBS and allow pathogens to then enter the brain tissue. That would not be good. And that brings us to the end of our first lecture on the brain. So just a little bit about what is there and how it's structured and how it's working together. Please let me know if you have any questions.